Is it online? You can't hear me? Okay, online people, you didn't miss much. I was basically saying whether we are alone or not, and how we're going to be talking about to what degree we can utilize experimental biology in order to reconstruct the past. The whole premise of our work in my lab is to synthesize and extract information from today's biology and today's planetary condition in order to infer ancient systems and early biology. Our planet is 4.5 billion years old. Life happened to this planet rather quickly. It's been evolving here for about 4 billion years. And the heart of the presentation today really hopefully will ask this question and maybe answer it to some degree. How does metabolic organization scale into an Earth-life integrated system? And to what degree that could lead detectable biosignatures on other worlds, right? So this is a scale problem. How do we connect what happens at the molecular level to a planetary level? And how do we then distill that information so we can detect that planetary level across distances, right? This is a major problem, of course, and how do we do this? And I'm very excited, honestly, to see the diversity in this room, especially given that we are trying to connect biology with astronomy. This doesn't happen, I think, to this level often. And the whole goal would be to connect molecular mechanisms to astronomical observations. So good luck to us all, because we're probably going to be doing this over the next century. All right, so if you don't know the answer to this, don't worry about it. That's why we're here. That's also why we're trying to synthesize this under research coordination networks as a part of the NASA's astrobiology program. And I think this question links LIFE, RCN, and Nexus really well. So let's talk about the problem here and where we come in. We have ancient metabolisms. The premise of this talk will be to understand to what degree we can reconstruct these ancient metabolisms in the lab, and how do they then translate into understanding the planetary biosphere, particularly detectable biosignatures. So I'm going to run through a little bit of problem, and I'm going to deepen the problem a little bit. Let's layer this problem into many, many other problems. It's like a wedding cake of problems, all right? And the first here is that we are largely unaware of how environmental and evolutionary innovations impacted our planet across time. This is even difficult to understand right now, right? How do you understand what a microbe does at the level of the planet today? Forget about trying to understand this at the 4 billion year scale. So this is a big problem, all right? Especially if you consider that we also don't understand the circumstances and the timing of events that made Earth a much different place. And I'm coming at this from a very biological angle. So let's review what these highlights are for once and for all. Of course, we are here because we love photosynthesis, but that's not the only major thing that happened to this planet. 4.5 billion years, hell like Earth. I'm very conservatively putting the first life, that's the last universal common ancestor. That is not the origin of life, but that's the first tractable life that we can at least certainly claim that exists and it was present on this planet. Say roughly 3.8, 4.1 billion years. And then I'm highlighting another big major event, that's the evolution of cyanobacteria, and what cyanobacteria invented that brought us all together here too, not just as species, but also as in this room. And then jumping forward, we have eukaryotes, and moving on, origin of green algae, animals, and land plants and surface ecosystems. So why am I choosing these, and why am I highlighting only a few handful examples? The reason is because these are not only connected to one another, these are also events that only evolved once in the history of our planet. And there's no competing or comparable example to these. So there's only one origin of life, one last universal common ancestor, one cyanobacterial ancestor, one oxygenic photosynthesis, one origin of eukaryotes, one origin of animals, etc. All right, so these are singular events. I like to think of these as evolutionarily singular events, right? One. And look at the gaps between them. These are large gaps, all right? But we don't understand how did they emerge. We don't understand why there is no competing reaction. We don't understand why at the molecular level, why certain metabolisms only also evolved once. And I'm going to touch some of these things in depth in a minute. But let's go to the second layer of this wedding cake of problems. There are major challenges accessing this magnitude of innovation. Again, keep in mind that when we talk about early life, we're talking about what we can track. That we should not assume that this is all there is. Our planet is covered with oceans, covered with forests. We don't probably have access to many other innovations, but this is what we can track thus far. When it comes to understanding life, and especially ancient Earth and ancient metabolisms, most of the systems that we study they originated during the first 2 billion years. And that's the best of times. The worst of times is also because this is a very largely intractable period, all right? There's little or no significant variability amongst taxa. You cannot really find morphological evidence when it comes to last universal common ancestor. We have some chemical biosignatures, which we will talk in a minute. But most of the innovations, I think we can agree, that happen at the molecular level, right? You're not going to get an actual microfossil. We're still debating about the cyanobacterial microfossils at the end of the day. Even though most innovations happened at the molecular level, they triggered big biogeochemical change and vice versa. So the molecular evolution was very much in touch with the biogeochemistry of the planet. And this is, I think, very much of interest for us, especially biologists. We should worry ourselves about what all these mechanisms and metabolisms do to the direct environment of the microbe and to direct biogeochemical environment, which I think is a bridge that biology, especially molecular biology, has not yet created. Geology did, geobiology does this, but biology doesn't come at it from a mechanistic point of view. So there's a big gap there. And of course, there are almost no direct remains of life from early Earth. That's another problem. But when it comes to biosignatures, especially ones that involve exoplanetary biosignatures, even though early innovations may not leave any fossils behind, they can leave certain biosignatures that could be detectable across space. So this is promising in a way, but again, look at all the other problems beneath it. So what do I mean by this? I wasn't here yesterday in case biosignatures were discussed. I'm sure that they were, but the earliest evidence of life, biosignatures, I'm highlighting just as I mentioned, there's only a few of these things, but if you look, if you go back further, you don't see the fossils, early fossils, but in a way, the crud that they exceeded. And you're looking at the early cyanobacterial fossils by Bill Schaff's paper here that dates about 3.5 billion years for cyanobacteria. We can debate for hours and hours about this, but when it comes to geochemical evidence, this is what we are limited with. And as I mentioned, there are gaps between these innovations, not only at the organismal level. When it comes to metabolisms, there's also singularity. And oxygen photosynthesis is one of them, and biological nitrogen fixation, for example, is another one. Carbon fixation is not necessarily a singularity because there are multiple pathways to this. That's my definition. I would be happy to hear your thoughts about this, but I would not define carbon fixation as a singularity. All right, so final layer of the problematic wedding cake. There are no obvious mechanistic ways to link the molecular variability on short time scales because we study molecular evolution. We know mutations happen really fast, but when it comes to biosignatures that we can detect at the planetary scale, they are happening every other 100 millions of years. All right, so there is some gap between these events. We don't know this. It's a miracle. Oh my goodness, I don't understand it. It's a curse at the same time that we understand that tiny molecular changes that can transform a planet, but the path is hard to reconstruct. All right, so what we need is a new way, I think, to study the past. We can agree that Precambrian innovations, that's about 4 billion years, has been majorly dominated by microbial innovations. And I will say that that's a molecular planet at that point, right? So past Cambrian, I think we can discuss the different level microscale evolution, but we are really talking about molecular evolution and metabolic evolution in particular when it comes to 4 billion years ran by microbes. So this is a microbial dominated billions and billions of years of time. And again, what we need to understand. My lab developed this concept. We framed it under the concept of planetary microbiology. What we are aiming to do is to reconstruct planet scale chemical consequences of microbial metabolisms, reducing this down to enzyme evolution. And we dissected the cell and life into 3 main components. Really, I think any planet you go, you're gonna get an interaction between information, environment, and metabolism. Information on our planet, genetic system, genetic code, etc. There's gonna be some information anywhere you go, anywhere in the universe. I think that will be coded. I don't even care what it is based on. There's going to be a metabolism. Electrons will have to be channelized in an organized manner across membranes, and it will be in touch with the environment. So this is probably the core universal structure of life across anywhere. And what we've done for now almost 8 years since I launched my own lab is identify certain innovations, again, as I mentioned, these singularities, but really focus on the molecular level behavior in order to ask two questions, and that's, I think, relevant for this group. One is that how can we use life as we know it to reconstruct ancient metabolisms on past Earth? And two, and that's what I'm going to be focusing on today too, use past Earth as an alien world and see what we can infer about light-powered life and ecosystems elsewhere. Of course, we are just a single lab, even though I'm also leading a NASA ICAR, a NASA center that looks into early life. I'm hoping that you all will find some relevance as to what we've done, what we created, and apply it to your work. All right, so how do we use life as we know it to reconstruct ancient metabolisms on past Earths? By the way, is this the first time you're mentioning life as we know it? Is it officially upside down? That's astrobiology's famous, favorite word. Well, what we do in my lab is a molecular paleobiology approach. We synthesize evolution. We start with phylogenetics, start by curation of modern sequences, create a lot of gene trees, create a lot of genome trees, and try to understand the evolution at the gene and protein level at first. We then move on to experimental phase. We engineer the genes, we infer using mathematics and statistics up until that point, but really create these genes synthetically and engineer them inside modern microbial genomes by replacing them with the modern counterparts. So in a way, we are asking this microbe to exist in the presence of an ancient DNA that comes from the past. So you may ask yourself, how will a single gene impact the entire behavior of the microbe? Well, not all genes are equal. Turns out some of them will express proteins that will dominate the entire behavior of the microbe that actually will be relatable to the biogeochemistry of that organism. And we've actually started this looking at metals. I'm going to talk a little bit about that, but the whole idea here is to connect the molecular record with the geologic one. And as I mentioned early on, I think earth sciences have been doing a great job with this. They have a better way of coming at biology. For some reason, biologists are hesitant to study the past in a way that geologists are. But I think with the tools of synthetic biology and artificial reconstruction, we can actually push this a bit forward. And I have a lot of opinions about why I think biologists stayed away from studying ancient Earth. I can talk about this for the entire week, okay? So we can talk about that. Anyway, so how do we resurrect ancient enzymes in the lab? We use ancestral sequence reconstruction. We like to think of this as molecular time travel. We start with modern protein sequences. We use many different evolutionary models. In my opinion, there could be better ones, but we're not model developers in my lab. We use currently existing ones. They basically assume how fast the DNA evolves in a current environment and assumes whether the presence of the amino acid in a protein sequence and the rate of evolution. Basically, that's what the model presumes. And we use multiple models and access ancestral protein sequences. I'm going to show a couple examples to dig deeper into how we're doing this, but this is for astronomers in the room. It's about taking the modern language and inferring an ancient one, all right, using DNA and protein sequences as a proxy. So where do we go from this? What we've done in my lab is to create this experimental framework where we don't only rely on, right, sure, great, we can do ancestral reconstruction, we can create these genes in the lab, but how do I choose which protein and enzyme to start with? So we decided that whatever we are utilizing in study needs to check these points. And I'm going to talk about an enzyme in a minute that's called Rubisco. We heard a lot about it. And Rubisco checked these marks. And what we're looking for is an enzyme that can be related to some sort of geological history that could be extracted from the rock record. Rubisco is great. It's a very dominant enzyme in terms of the metabolism that it participates in, and the outputs of this metabolism, carbon fixation, can be extracted from the rock record. It is characterizable, it's geologically relevant, and there are appropriate modern hosts, like an engineered cyanobacteria, to study Rubisco, which is also relevant for early life. I mean, you don't want to engineer spinach if you're interested in deep time, right? So these are interesting, I think, and relevant nuances that one needs to consider before choosing the enzyme to study when it comes to relating to ancient Earth. Since then, we moved on to nitrogenases. We're working with nitric oxide reductase, so I know there's some interest in the exoplanet community about these enzymes, but we basically study many, many different enzymes that look at different time points across time. Here I'm highlighting the first roughly billion years, and all the different enzyme systems that we tackled over time, looking at origin of life, as well as the early information systems. And what do I mean by the importance of linking the enzyme behavior to rock record? Is that great that you're looking at a microbe, in this case, this cartoon is depicting what a living microbe will generate in the rock record in in the particular example, looking at the carbon fixation and the carbon isotope biosignatures, ultimately what determines this is the enzyme and is enzyme's activity. Right? So what we realize is that we can compare an ancient carbon isotope biosignature to a modern existing organism, but we are not really integrating evolution into our understanding of these records, right? We are not thinking about how did the Rubisco evolution factor into the generation of these chemical biosignatures. We started with that. I'm going to skip this rather fast. As I mentioned early on, there's going to be a few rapid fire examples, so stay with me, please. We heard a lot about Rubisco, so I'm going to skip the introduction. It's humongous. We love it. We hate it. There's just a lot of emotions around this enzyme in the community. But we generated the evolutionary history of Rubisco. This is where we started. This was the launch of my lab as well. We ultimately developed an engineering protocol for cyanobacteria where we excise the modern Rubisco components and insert the ancient Rubisco genes inside the genome. We published this. Oops. Whoa. And then we basically calculated the carbon isotope fractionation of cyanobacteria that now creates this ancient Rubisco component. Right, so we were able to combine synthetic biology with geochemical isotope probing, and I think hopefully created a whole new area of study where we can perturb genomes and link them to paleontological records by way of studying the isotope outputs of these engineered enzymes. When it comes to Rubisco and carbon isotope fractionation, it wasn't mind-blowing in the sense that we were able to demonstrate that the uniform carbon isotope fractionation record was indeed maintained despite the evolution of this enzyme. The record is persistent, and we can recapitulate whatever is extracted from the rocks. When it comes to carbon, we moved on at the same time. We looked at biological nitrogen fixation, another singularity. There's only one way life invented, how to utilize otherwise unavailable nitrogen in the atmosphere in a form that is available to life. We tackled this quite a bit. We're still working on it. It's what we like to think is a good case for planetary enzymology. It relates directly to the biogeochemical evolution of the planet as well. And this is the enzyme that runs the show, as I mentioned, just like Rubisco. This is also a very dominant enzyme. Without nitrogenase, there is no nitrogen fixation. All right, so these are very important players. We started by looking at the evolutionary history of nitrogenase. I'm going to skip all of that because you can find all those papers, but I will highlight a recent study that we've done. And Holly Rocker, she's actually going to be talking about setup cyclone, where we resurrected the ancient nitrogenases this time and engineered a nitrogen-fixing microbe, a diazotroph, at the genomic level and measured the isotopic output of this bug in order to recapitulate the ancient nitrogen isotope record using living organisms. And what this enables us next step is that we want to animate, so to speak, the environment and see if we can mimic not only ancient Earth, but different planetary bodies and see to what degree we can vary the isotope generated by these engineered enzymes. So we basically want to put an exoplanet in a flask and see how these microbes will behave as we change the environment and the food that they survive on. So I hope you have a chance to visit her talk. She's a great speaker. Well, another interesting thing about nitrogenase, and I know many of you here are love enzymes that eat metals. That there's a lot of people in this room who studied this, that metal concentration across our planet did not stay the same. Of course, through the oxidation events, there's a big revolution, but regardless, the metals have to come from somewhere. So biology, right? Earth provides, biology demands, and we have to think about when we study our beloved metabolisms or photosynthesis and all these other enzymes, where does this cofactor come from, and what happens at the scarcity, potential limitation of this cofactor? So we started with nitrogenase. It's a metal-loving enzyme. It has a molybdenum, a vanadium, an iron version. We simply asked what is the ancestral state and what does the ancient enzyme prefer. Just to keep the story short, because we published this briefly. I'm sorry, we published this, but I forgot that I did this, but I just wanted to point out that this paper just came out in annual review of reviewers of Earth and Planetary Sciences, talking about the bioinorganic bridge that is very famously linked biology and geology together. So 20 years later, 25 years later, we are revisiting this bridge in order to understand how biogeochemistry and biology can be reintegrated. It just came out, so please take a look if you're interested in metal evolution in general. But what we have done is not only look into molybdenum dependence and nitrogen fixation, but all molybdenum dependence across life. And we created a survey of all the enzymes that depend on molybdenum as well as tungsten, because these, if you don't know what molybdenum is, that tells you something, right? I'm sure we all heard about iron and the importance of iron, but I always say, oh, I had no idea molybdenum was so important, because it's a rare metal, but the entire nitrogen fixation, for example, depends on this metal. So we mapped the complete molybdenum pathway of the cell and basically tackled the age of biological use. And what we found is that molybdenum, even though it was extremely limited in the environment, was actually already utilized by biology as early as about 3.6 billion years, right? So these rare metals, despite their limitation in the environment, they are preferred by biology. So we learn in biology that the environment selects, and to some degree that's true, but just because something is limited in the environment doesn't mean life is not going to build upon it. But what does this mean when it comes to life search? We always talk about, are we looking for life in the right or wrong places? And where should we look for metal-enabled life? Yes, we cannot put a lab on an exoplanet, but I think we can distill some sort of observables and tackle, maybe measure and develop some measurements using a limited set of tools. We understand that for nitrogen fixation, once the molecular architecture is present, metal requirements become a constraint. So you have to understand the origin of these metabolisms, and that's very important. But signatures that are critical for biology will depend on, in most cases, the presence of these cofactors and elements in the environment. And how much metal does ancient life need? How much metal is needed for the basically jumpstart these metabolisms? So, which one? 2 or 5? 7. That we need to. So for this, we went ahead and did an analysis of the transition metals in low-city hydrothermal fluids. And this was actually done by one of our collaborators in our ICAR, Bill Safe Foods at University of Minnesota. They have this amazing experimental tool where they can generate a hydrothermal system in a lab with enough pressure and environment and temperature to understand basically the metal requirements around life. In addition to creating a lab system, they also again measured the actual fluids from these vents. And on the right, on top right, you're looking at the amount of vanadium, iron, cobalt, nickel, molybdenum, and tungsten amounts that were calculated in the low-city vent fluids. That I think clearly shows that hydrothermal systems can concentrate the metals that life needs. Even though a planet's ocean may look globally metal poor, what matters is whether these concentrated environments can actually support these metabolisms. And I think, even though I think most of us have issues with the word habitability, because I don't know what it means anymore, but it's not only about average planetary composition, but about whether planetary processes can create local chemical niches where metabolisms can emerge. So that's going to be important. Very fast, moving on to point 2. How can we then use past Earth as an alien world to predict, in this case, light-powered life in ecosystems elsewhere? Just please recall that Earth life is an integrated system. Our planet is very much living. I don't think there's anything called abiotic on this planet, actually. And so the earliest proteins, we don't know exactly what they are because we never truly evolved a protein from scratch, a protein that we understand. We don't know the origin of any enzyme yet. But we can be, to some degree, agree that especially this sort of photon-powered enzymes, looking at the simplicity of their structure, could likely be one of the earliest enzymes to evolve on our planet. So the question is then, can these ancient photoproteins reveal where light-powered life could live on early Earth and other worlds? So with this, we yet created a different synthesis this time. Let's not only look at the behavior of the protein, not only at the functional level, but also at the geochemical level, but can we also connect this to astronomy? So this is a completely schizophrenic astrobiology study, okay, where everything comes together, because I'm not sure any paper that actually linked protein evolution to astronomy, then these measurements were done by Eddie. So using ancient light harvesting proteins, our goal was to look at whether we can find habitable niches. We looked at microbial rhodopsins in this case. These are simple ancient life-powered proteins. They are believed to predate oxygenic photosynthesis. They're possibly one of the earliest light harvesting systems on this planet. They have remarkable ecological versatility. They are evolved across very diverse environments, and they have an adaptive spectral tuning property. Different rhodopsins absorb different light, green, blue, or even UV light. And so they enable microorganisms to occupy different ecological photic regions. The goal was, again, to tie together bioinformatics, geochemical, and stellar analysis. We started with what we do, create all sequences, align them, and infer the ancestral sequences across time. In this case, because we presume this enzyme to be as old as life, we think the oldest one we analyzed is about 4 billion years old. I don't know any study that truly dated retinol-based proteins. Maybe this community will do it. If I'm missing a paper, please let me know. But I wanted to mention that there's a lot of machine learning going on behind the scenes when it comes to these protein analyses. We're not just taking one sequence and running with them. We do hundreds and hundreds and hundreds of different ancestral protein reconstructions and calculate all the posterior probability metrics for each site at the protein. All right, and then we create these massive libraries, sometimes hundreds and thousands of proteins. And what served us well in this case is that there was another machine learning algorithm developed by our colleagues in Japan that functionally also predicted the outputs of these functional predictions using machine learning and AI. So we could predict the actual wavelength and the light emission of these enzymes using machine learning algorithms. We created a lot of different alternative ancestors, but the take-home message is that the most ancient rhodopsin were tuned for green light, which was consistent with the light conditions of shallow marine environments. So we're looking at the enzyme, and we're trying to infer the residence and the ecological niche of the microbe that hosts this enzyme. All right, and I'm just going to read this together with you. That again, ancestral rhodopsins are green light absorbing ones, and we think that after the great oxidation event, changing planetary light and redox conditions may have actually triggered the diversification of rhodopsins into new niches. And Ellie is in the room. Please see her poster for more on this. But what we've done here is that, and this is the part where there's astronomy's broken dreams, right? Where we actually tried to look into what sort of habitable light environments can this protein reside in by looking at, in this case, this is what Elish Widerman did. The question we asked was, given the light environment of early Earth, what kind of light harvesting biology could live there? In this case, we combined our own results with the Precambrian Earth light conditions and modeled basically different conditions of the planet starting from the top. We were looking at modern Earth, Archean, as well as the water transmittance and normalized absorbance. And with that, we were able to probe the ecological niche that this microbe that is presumably hosting this ancient photon protein is residing. That was basically closer to the surface. Take-home message here is that Earth's atmospheric evolution has drastically altered the surface photon radiance profile over billions of years. This is good news, I think, for protein modelers. Why not think about ancient sun and the light attenuation when it comes to understanding protein evolution as well? It's time we bridge these things. And the take-home message here would be that I think, and I will say it, don't boo me, but instead of assuming that alien phototrophs use only chlorophyll or oxidative photosynthesis, we should ask what wavelengths are available on the planet and what simpler light. So light harvesting systems like rhodopsins and maybe even simpler enzymes could also evolve there. I added that also to soften my statement. All right, so take-home messages from part one. Ancient metabolisms, I believe, provide a universal marker for detecting life beyond Earth. We need empirical information. This is good for biologists. We should be able to constrain the planetary chemistry that makes metabolism possible, even though I understand that when it comes to exoplanets, we don't know much about the planetary chemical conditions yet, but we will get there. And number two, different planets and stars provide different light environments, and biology elsewhere may not necessarily look like what we have here. And this is also great news because with artificial biology and synthetic systems, we can create a lot of alternatives. And forget about experimental systems, we can do this at the computational level too using AI and all. So always consider the alternative possibilities. And with that, thank you so much.
Two quick questions.
Thank you so much. I really enjoyed your talk. I'm Sally from UC Riverside. My question is that when you do ancient sequence reconstruction and put a single gene or a few genes into a modern bacterial organism, that's still a modern cell. While an ancient organism could have an entirely different networks of co-evolved genes or regulatory systems. How do you see this mismatch? Do you see the field moving towards reconstructing an entire, network of metabolic pathways, or?
Yeah, that's a very good question. I mean, it's fair, right? So we're not claiming that we time traveled and collected IVISH, right? So it's a truly ancient microbe, right? So yes, in this case, as I mentioned, we do try to target enzymes that dominate the behavior of the organism, and you got to be careful with what conditions you're testing with this organism. I didn't talk about this, but we've done pretty much all of these enzymes under also biochemical conditions. It's cell-free. Ali, I think, talks about it a little bit. We are developing protocell systems right now, so we have our own little artificial cell. So we are expanding. We're completely aware and agree with you, but we have to start from somewhere, so that's where we are. I think some people are hoping that we will be able to create last universal common ancestor experimentally someday. Why not? I think we should do that, but not my group anytime soon.
This is just a really quick comment, and don't take it in the wrong tone.
Oh, yeah, oh my God. I will do it. Not that you said it.
No. So there's this sense of the challenges of exoplanets because we're looking at spectral data from chemistry, right?
Yes.
And how impossible that is in early Earth if only we had that. But you showed 3 signs of archaean life, and probably none of those were life, actually. The 4.1, those are carbon isotopes of graphite in the zircon crystal. Who knows, right? The 3.7 stromatolites from Greenland, probably not.
Really?
The Schopf microfossils, probably not. Certainly not cyanobacteria. You have to go to 3.4. The point is that you can fill a truck full of samples and take them back to the lab, and you're still mostly stuck with chemical analyses and genomes, thank goodness. But mostly you're stuck with that. And in exoplanets, we're just doing spectra of those kinds of things and trying to calibrate them by our understanding on Earth. But it's not easy even on our own planet to do this.
Thanks for the truth bomb. But what's the most reliable biosignature that you believe in when it comes to archaean?
It depends on what time period. I mean, the gold standard are organic biomarkers, which has driven much of the Mars research. But that's not going to work on early Earth in the Archean, and the Proterozoic it starts to work around 1.8, and after that, and there's still giant contamination issues. So that is amazing.
Because I have problems with the biological inferences of.
It's describing the particular organism. Sure, that's a whole other question, but maybe you wouldn't debate that they're life, right? But yeah, I'm a big fan of isotopic and elemental proxies for metabolisms, but almost everything we do on Earth has an abiotic pathway as well. It can be something similar. So it's all about context, and that's going to be the same thing on exoplanets. I always say that those are people in my world that eventually the data are going to come, but they're not going to be anything until a collective group like this can interpret them.
I agree.
Because there's so many ways that they can go. Thank you for letting me soapbox that.
Get it out of your system. Yeah.
Let's thank our speaker again.
Let's thank your speaker. Yeah. All right.
Our next speaker will be Massimo Maffei from the University of Turin.
Thank you. Can you hear me? Yes. Okay, let me thank the organizers for giving me the possibility to talk about magnetic fields, but I will talk about hypomagnetic fields, so not a radio frequency magnetic field or high magnetic field. So my intent is to focus on this subject, so the geomagnetic field, which is a natural shield from preventing erosion of the atmosphere, but also protecting life in this planet. And this is not a common situation in the solar system. For example, Moon and Mars, they have no magnetic field. So my topic of the research is to understand what plants will do when we'll start growing plants on the Moon first and maybe later on Mars. So this is my topic. And this can also give information on something that could be important to check for habitability of other planets. So in order to do that, we built this almost giant triple Helmholtz coil system, and it is continuously controlled by a flux gate. So we have the real-time measurement of the magnetic field that we reduced. This is the experiment typical of 3-4 days. There is no way to keep it constant because the magnetic field continuously changing in our planet. And so if you can download an app and see how much is fast the changing in the geomagnetic field. But we did a digital twin model with the CNR in Milan. So they were able to give us the possibility to understand how is the distribution of the geomagnetic field inside our system. So as you can see, we have about a sphere that is 30-40 centimeters in diameter, where we know exactly that the geomagnetic field is reduced at least 1,000 times. So this is enough space to have a plant growing like seed to seed in Arabidopsis or small plants. So we did a lot of work on this. I have no time to go through all of this, but starting from the involvement of geomagnetic field reversal in evolution, then we could demonstrate a delay in flowering time and the involvement of photoreceptors and mineral nutrition, the difference between root and shoot in perceiving the magnetic field, the homology of the genes that are present in birds, so in migratory organisms that are able to perceive the magnetic field, and there are homologs in plants and many other organisms. And then we did the Ross involvement in magnetic field changes, photosynthesis, of course, and the fact that the proton pump is affected by the magnetic field, in particular 1433 proteins do not bind anymore, and then many other things. That we also combine the effect of simulated microgravity with simulated magnetic field and the regolite use. So we just published last week the effect of simulating a plant growing in Mars gravity, Mars magnetic field, and Mars regolite. So just to give you an example of what the reduction of the magnetic field can do, here you can see transient fluorescence analysis, and you can see that the good news is that the pointer. The good news is that you do not have variation in FDFM, so the quantum yield is quite constant. There is no damage on the photosystem too. But as you can see, there are some other parameters that are changing. For example, you have an increase in basal heat dissipation flux per active reaction centers, or you have a decrease in what is the dispersion of temperature. But what is important is that you have some sort of congestion of the reaction center, and this is agreement with our observation at the ultrastructural level on the thylakoid membrane. So it seems like there is a reorganization of the reaction centers, and when you do NPQ analysis, you see that you have a clear reduction in the number of reaction centers that are open, and also the so-called vitality is going down. And so even though the plants try to react to this condition of growing in low magnetic field or in normal magnetic field by compensating a little bit with non-photochemical quenching, they cannot prevent what is apparently a traffic jams of electrons. Another thing that we did was, okay, we've been growing plant continuously in low magnetic field or in normal magnetic field, but what happened when you move this plant quickly to the other conditions? So the red line is the plant that is growing in a normal magnetic field, and here is 10 seconds after we just take out the magnetic field, and this is the reaction that you see. So in increasing in fluorescence, increasing fluorescence, and it's a very rapid reaction, milliseconds, you can already see something. But if you do the same on plants that is growing in the hypomagnetic field, and you put it back to the normal magnetic field. That plant already has built a new system, a new organization of the thylakoid membrane, so it cannot react as in the other system, and you see a decrease in fluorescence. The same can be done with the NPQ, so you see that in this case, once again, the plant that is normally grown, and then you take out the magnetic field, you see, for example, an increase in QE, but this is not the same when a plant is adapted to low magnetic field. So we need to find some explanation for this very quick and fast reaction. They cannot be justified by gene expression or reorganization of membranes or so on. It's too fast. So it's not a matter of energetics, instead it's kinetics and spin dynamics. I got every time a comment from reviewers, oh, the GMF is millions times smaller than the thermal energy, so it cannot work like this. But in fact, the GMF does not need to overpower the KBT, it's only need to influence the direction of the spin gate. So one of the explanation of this can come from quantum mechanics. So you can some sort of reconstructing the Hamiltonian of these radical pair interactions. So in the first term, you have the Zeeman interaction, which is dominated by the magnetic field, and then you have also a way to quantify this. You can put in your equation the Bohr magneton, but also you have to consider the electron response to the magnetic field, which can be slightly different between P60, 618, and ferrofectin, and also the electron is intrinsic magnetic moment. Then you have the other term, which is called the hyperfine interaction. Here, it is dominated by the hyperfine coupling, which is some sort of a glue that is keeping, let's say, the electron and the nearby nuclei, like in a tank that is coordinated, and then you have also the nuclear spin. Of course, you have to consider both of them. So the first term consider the two electrons, one for each of them, in order not to have a very long equation. And you also have to consider that you have to calculate in the hyperfine interaction what is the contribution of all the surrounding atoms with the electron. So what happens if you take out the geomagnetic field, then you take out the Zeeman interaction, and you leave only the hyperfine interaction. And this could create problem in the equilibrium between the singlet and triplet state, for example. So what we think that is happening is that in normal situation, what you just saw the timeline of life. The geomagnetic field was formed before any kind of life in this planet. So life evolved in the presence of the geomagnetic field. And in this normal situation, you have what is the tug of war between these two components, the Zeeman and the hyperfine, but it's perfectly balanced. So in normal situation without stress, you have the singlet state that is dominating, and then you have transfer of electron. But in this situation, when you take out the magnetic field, you have some sort of, let's say, congestion of electrons. You have a decrease in photochemical quenches. You have an increase in the QE. And then it is like, let me give you this example, having a rock band. And in this rock band, they are playing, but at some moment you take out the drummer. So what happens is that you have only the instrument, so the hyperfine, that is playing, but you are losing the rhythm. So you have no way to keep it constant as it was before. What we are doing now, we are working on several mutants because we want to understand what is the real reason of these changes in the non-photochemical quenching or in the OIJP forms of fluorescence. So we are using different mutants in order to try to avoid, for example, violet xanthine and zeaxanthine interaction with for example, the triplet that could accumulate, or the PSPS structure in order to see whether or not it is involved. But since we notice a QE increase, then we might think that it could be something that is related to a change in the delta pH, or whether there is an involvement of, for example, the singlet oxygen. Then we are using this mutant that it is overproducing this singlet oxygen, and we are also working on these two mutants. First, because this is the homologue of the magnetosensor in birds, and then we have a KO and overexpressing mutants that we are working with. But also, it was interesting to note that if you have an overexpressing, an overexpressing cryptochrome or downregulated cryptochrome, they interact oppositely on the NPQ. Very, very quickly, the take-home message, when we will grow plant on Moon, on Mars, of course, we are not growing plants outside, so we will grow in greenhouses. Probably these greenhouses will not have the same atmospheric pressure, but the plants will feel the lack of the magnetic field of these places and also the gravity of these places. So we have to find a way to understand how, through synthetic biology or targeted acclimation, we can just modulate those plants in order to stand the new situation of these conditions. So thank you very much, and sorry for the delay.
Okay, one quick question while we're switching over. Anyone have a question?
I feel like I'm the geologic police here. Somebody sent me an article about why is it that geologists are always the earliest characters to die in movies. We don't know about Earth's magnetic field as well as you described. John Tardoon, I won't go into details, but there was a claim of 4.2 from zircons. That's been challenged by an MIT group, and anything before 3.5 is really pretty unknown. I suspect that there was, but we don't really know. And so we can't go forward imagining all these different scenarios under a well-established magnetic field in that early part of life's history.
Yeah.
We just really don't know.
Yeah, also there is the formation of the moon that probably contributed more.
Oh, that was a long time ago.
Yeah,
was 4.5.
Yeah. Yeah. Anyway, there are reversal in the geomagnetic field, but this cannot be traced more than 200.
Well, maybe the way it's to flip it and say that this is required by a magnetic field. It's early, and therefore there must have been a magnetic field.
Yeah. Anyway.
Thanks.
Okay, let's speak again. Our next speaker is Kenji Takezawa from the National Institute for Basic Biology in Japan.
Hello everyone, this is Kenji Takezawa from Agouron Center in Japan, and I'm also drawn to the Basic Biology Institute. But our institute, Agouron Center, is established 10 years ago for studying the habitable exoplanet and the life on there. So searching for life on exoplanet, phototropes like a green plant is the best candidate for detection, because the oxygen photosynthesis produced important biosignatures. The first one, primary one, of course, the oxygen and also ozone in the atmosphere. And the secondary biosignature is the so-called vegetation leadage. So in general, green plant leaves shows the specific characteristic pattern and light reflectance. And those features comes from the two important features of the photosynthesis. One is the thick rich tissue that's scattering the light in wide wavelengths, and that's increased the baseline of the light reflectance. And the other is the specific light absorption because of the photosynthetic pigment. In the case of the chlorophylls, the light absorption is very strong in the visible light visible light, especially blue and red light. So these two features create this unique light refracting spectrum, so-called vegetation red edge. And that signal could be detected remotely and used for the indicator of the life on the exoplanet. But the question is that the wavelength position of the red edge of the vegetation could be shifted depending on the light condition of the planet. So most of the habitable planet for such the life is opting around the M dwarfs. The temperature is lower than the solar sun, and in that case, the radiation is shifted to the near infrared region. And there is only the small amount of the visible light is available on those planets. So many people think that if the light condition is different, the plant color, leaf color is different. And in that case, the light exposure should be different. So using the different light for photosynthesis, there should be the different mechanism to utilize the light energy compared to the chemical energy useful for plant and animals or other creatures. So let's review the Earth-type photosynthesis. So Earth-type photosynthesis is driven by the two-step sequential energy input in visible light. And that amount of energy required to transfer the electron from the water to the reducing reagent against the redox potential. So for use the NIR, near-infrared radiation, instead of visible light, this system that cannot be used because the single photon energy and the near-infrared radiation much smaller than visible light. So there are many hypothetical mechanisms enabling the reaction using the near-infrared radiation, and I show here the three major ones. And could be the most popular one is the multiple reactions. So that's the not the two sequential, that's the three-step or four-step of the reaction. And even if the single-step energy, achieved energy is smaller than a visible light, the total amount of energy could be equivalent to the two-step reaction by the visible light. The other one is the second one is the energy conversion, conservation, like so because the certain amount of energy is lost during the electron transfer reactions, even if the input energy is smaller, the loss of energy is if we can improve the energy efficiency and reduce the loss of energy between two reactions, the final redox potential achieved at the same level. And the third one is the two-color reaction. So this is the easiest one. The first step is the same, using the visible light, and the second step is the near-infrared radiation. So which one is the possible for on the M dwarf is the question. So summarized, I compared the light environment and on the hypothetical habitable planet around M dwarf, around AD layer, which is the M dwarf star. So this panel shows the light intensity spectrum on the habitable planet around the AD layer. So green line shows the light intensity on the land surface because there is a lot of near-infrared radiation and less visible light. But the situation is very different under the water. So this three line, you can see the color, but the red, green, blue shows the light intensity under the 10 centimeter, 1 meter, and 10 meter depth water. So under the water, there is only the visible light. So the situation is very simple. Even if the light intensity on the surface is predominantly in a near-infrared region. Under the water, there is the very similar condition between the planet around the M dwarfs and the Earth. So we can estimate that first life during photosynthesis underwater should be the same type of photosynthesis to the Earth. So let's begin the Earth-type photosynthesis and consider if it is possible the evolution from here to here. The answer is no. Because Earth-type photosynthesis is two reaction center is originated from single common ancestor. So that it has the common partner to pass the or accept the reaction and easily constructed two sequential mechanisms. But it is very hard to put in one more. And the easiest one is the two-color reaction use the visible light for the first step and the second step to the near-infrared radiation. But this one also has the difficulty under some condition, especially the light spectrum is different because the two reaction centers use the different light. It is very hard to balance the excitation. And that's especially difficult under the shallow water. In the shallow water, the ratio of visible light to near-infrared radiation is drastically changed with just 1 meter depth water. So actually the green clouds in the Earth has the very sophisticated mechanism to balancing the two reaction centers called the state transition, which is the shuttling the light arresting antenna between two photosystems. So I calculated if this system can work under the M-dwarf's water. The answer is no, because the difference in the spectrum is so big, and to balance the photosynthesis system, almost all peripheral antennas should be shuttled between two reaction centers, and it's unreasonable. And even if it's achieved, the increase in the light LF is less than twice. So that's not very beneficial. And the conclusion is that around M-dwarf, there's a lot of radiation, but in water, only visible light. And there is the Earth-type photosynthesis using visible light is both first, and it keeps using visible light until completely adapt to the land surface condition. Yeah, that's it. And thank you. I'm happy if you have any questions.
We have time for questions. Any questions? So, what M-dwarf did you assume? M-dwarfs span a range of effective temperatures. Was there a significant difference between, say, a late M-dwarf and an early M-dwarf?
You mean in temperature?
In the
Spectrum. It's a spectrum. So we estimated that the young M-dwarf is very active, and there is a lot of the flare, strong UV radiation. So that's the reason we estimated that the first phototrophs evolved in the water, at least 10 meter or a few meters depth.
Yeah, I was just going to mention that the likely HWO target list includes only 3 M-dwarfs whose name and addresses were known to 1800s French astronomers. And so it might be good to focus on those particular nearest objects when modeling the M-dwarf reflected light signatures.
Yeah, I agree that, but basically the conclusion is difference to choose any type of M-dwarfs because the character absorbed of the water molecule is the predominantly factor to decide is the light environment in the water. I tested the black body radiation spectrum and can can reproduce almost the same result.
But on the more optimistic view, the ELT is going to focus a lot on the M dwarfs. So no need to restrict to those three.
Let's thank our speaker again. Our last speaker for this session will be Greg Fournier from MIT, who will speak about mass extinctions of phototrophic lineages.
And I have the enviable position of being between everyone in the bar, so yes. Thanks for the invitation to talk here today. I think this, what I can contribute to the conversation the last couple of days is, I think, a completely different kind of information, but I'm hoping that it's complementary to a lot of the other talks from different disciplines that we've heard so far. Today I'm going to talk about molecular clocks, phylogenetics, and the evolutionary history of a really important lineage in the history of life on Earth, Prochlorococcus, as a case study for thinking about the types of successions that can occur and what they can tell us about habitability and the potential for even detecting inhabited planets elsewhere in the universe. My talk has 4 short sections. First, I'll talk a bit about Prochlorococcus, what it is, why it's a great model organism for thinking about these kinds of things. I'll establish a bit of background on previous work that's been done, trying to estimate how old this clade is. I'll talk about some of our newer methods and the age estimates we're getting now. And then the impact not only for the particular question of how the evolution of this group relates to one particular event in Earth history, the cryogenian snowball Earth, but also what that can tell us about marine microbial habitability in general. So first of all, Prochlorococcus. This is the most abundant marine cyanobacteria in the world. It's prevalent in equatorial waters. You can see this. This map was done by my colleagues at MIT, where you can see the green is Prochlorococcus, so it's predominant in equatorial waters. And then the related marine Synechococcus are at higher latitudes. And then eukaryotic phytoplankton dominate in polar regions. But this is the most abundant photosynthetic organism on Earth, responsible for half of global oxygen production and marine CO2 fixation. They're really tiny and they're really weird. They have a streamlined physiology, streamlined genomes, and they have specifically adapted in many ways to the really harsh, nutrient-limited conditions of the open ocean. This is the largest surface niche on the planet. So why am I interested in this particular clade? So because it seems unique in terms of its strange adaptations and overall impact on global biogeochemical cycles, we can ask what conditions triggered or permitted the evolution of this group of cyanobacteria in this particular niche, which is out in the open ocean, not just typically more productive and more nutrient-rich coastal environments. Historically, this has been tied to hypotheses about the cryogenian snowball. So, did marine picocyanobacteria, that's the group Prochlorococcus belongs to, together with marine Synechococcus, did they survive the cryogenian snowball Earth interval? I mean, if that's true, then yeah, this niche is robust. Like if snowball Earth can't even kill, equatorial warm temperature cyanobacteria, then, these groups may be very persistent across long geological timescales, regardless of the changes on a planet, which would suggest that there's persistent, robust habitability, and these things are really hard to kill once they get established. Or did they evolve right after the snowball, which would suggest that they're part of a recovery, and then maybe there were groups doing this before the cryogenian, and then they got wiped out, and then what we're seeing is the most recent adaptive radiation replacing it, kind of like the new types of corals that show up after the end Permian mass extinction. Or did they have nothing to do with the cryogenian, and maybe they evolved much more recently due to other adaptive or selective pressures, and really the narrative is not one of long-term habitability, but simply the opportunity that presents itself as these niches co-evolve with other life on Earth. And then in general, how sensitive would these groups be to major climate changes, which become very important if you consider the multi-billion-year history of the planet. So a lot of work has been done previously trying to establish this. I am not going to go into the detailed workings of molecular clocks because of time, but needless to say, it's very hard to date the evolution of microbes because they have next to no fossils, and there's very little information we can use otherwise. Some groups produce lipid biomarkers that can be diagnostic for some clades. That's helpful, but oftentimes they're not diagnostic for particular groups, and that record is also very scant when you get into the Paleoproterozoic. And this is extremely hard, especially for Prochlorococcus, because it doesn't have any diagnostic lipids. It lives out in the open ocean where even if it was leaving fossils, those get subducted over hundreds of millions of years. It's not in a place where you would expect preservation to occur. And because it has a weird genome composition, it's evolving really fast. And so it has long branches and strange compositions that really interfere with accurate molecular clock studies. And because of this, it may be unsurprising that age estimates for this group have varied tremendously in the literature. So I'll go through some of these very briefly. Most recently, and this got a lot of attention, so you might have heard of it, was age estimate by Zhang et al. That suggested that Prochlorococcus has a group diversified in the Tonian and survived the Cryogenian, and that its evolution, they directly implicated the evolution of Prochlorococcus as maybe a driving factor for putting us into the snowball. Another analysis, and this is similar to several other, this is work done by Patricia Sanchez Baracaldo, and these ages are similar to what has been gotten by like Patrick Shee and Woody Fisher and ourselves in previous analyses, is a Phanerozoic origin for Prochlorococcus and marine Synechococcus. Which would suggest that it is not implicated in causing the Cryogenian snowball and did not survive that bottleneck, but represents novel diversity that emerged afterwards. The most recent work that our group had done with this in collaboration with Penny Chisholm's group is identifying enzymes within marine Synechococcus and Prochlorococcus that also bind and degrade chitin. And this gave us another hint that we're looking at a Phanerozoic phenomenon because there wouldn't be a lot of detrital marine chitin in the Tonian. And so integrating this with our previous molecular clock ages that you see up here, which these age intervals are quite broad, but suggested Prochlorococcus and marine Synechococcus are evolving in the early Phanerozoic. So we developed this rafting hypothesis that there was a stepwise transition from coastal environments to the open ocean using particles of detrital chitin that the ancestors of Simpro would bind to and be this intermediate to raft out until they could evolve to be completely independent, living in the low nutrient environments in the open ocean. And this timing works out with the evolutionary history of marine arthropods that produce chitin. But these age ranges were still highly uncertain. And we're still left with that disagreement. Why do we get ages so wildly different from other studies? And if you look at these ranges, you can see the problem. So we have the Zhang et al. Work going all before the Cryogenian. We have our study. We have different studies by Sanchez Baracaldo. And the uncertainties are huge. And it becomes very hard to implicate any particular causal mechanism when you're looking at these large uncertainties. And so, and the reason why the uncertainties are huge is if you think of how molecular clock works, we have the problem of a distance between the fossil calibrations in the group we're interested in. So fossil calibrations, if they're close to a group you want to date in the tree, are going to be very active in constraining the age, and evolutionary rate models will have to accommodate those constraints. But when you're dating Prochlorococcus, your nearest calibrations are are billions of years away. They're a handful of groups of cyanobacterial fossils, some of which might be dubious, but more importantly, it's the plastid, the lineage that gave rise to plastids, and those eukaryotic organisms that contain them, their fossils are then used to date cyanobacteria. But you can think of it, if you're a sedimentologist, you can think of the problem like dating with an ash bed. If you have an ash bed and then you observe fossils right above it, that's a pretty tight constraint if you can date that ash bed. But if you have a huge amount of sedimentary layers between the ash bed and the fossil, then the age you get will be entirely determined by your eighths of the rest of the rate of sedimentation. And the bigger that stack is, the more and more uncertain it will get as the rates can vary over larger intervals of time. And that's what challenges molecular clocks. So what we tried to do is solve this problem by coming up with novel age constraints by using horizontal gene transfer. So I'll skip this. It's if you're interested, I can talk about this more afterwards. But basically, what horizontal gene transfer does is it allows us to connect the evolutionary history of a group without fossils to the history of a group with fossils. And so then we can proxy date the lineage that does not contain fossils by the constraints imposed by acquiring or donating genes to groups with fossils. You can think of these as cross-cutting events. And we found 3 gene transfer events that are very powerful in constraining the potential ages of Prochlorococcus. So one of them was a transfer from within crown green algae to the ancestor of all picocyanobacteria. And so given the fossil record of green algae, that constrains this node to being between about 750 and 550 million years old. That's running a molecular clock just on this gene tree. Another gene transfer done by Feichi Inama in our group. Protein has no known function, but fortunately function doesn't matter for this. And we see all of these different eukaryotic groups that all have different fossil records, dinoflagellates and diatoms and brown algae and green algae. And a gene transfer to Senecacoccus prochlorococcus, so a more recent event. And a molecular clock constrained by all this rich fossil record gives us an age for this split of between 689 and 500. And then another gene reconstructed by another grad student in our group, Maya Krager, fatty acid desaturase. We see the same thing. Lots of eukaryotic groups on this tree, and then at some point a gene transfer to Prochlorococcus, which gives us an age constraint of 686 to 441. These are all totally independent, using different genomic information, different phylogenies, and in many cases, different eukaryotic fossil records with dozens of constraints, but they're all giving us ballpark similar ages. So if we throw these ages onto a species tree of Prochlorococcus using many genes, so we can get a more robust species tree and a better posterior age distribution on our clocks, we can get new ages for Prochlorococcus. And when we do this, we see crown group Prochlorococcus evolving in the Ordovician. So not back here and not right after recovery, and even marine Senecacoccus prochlorococcus look to be evolving in the Ediacaran sometime after the Cryogenian. And what's nice is this resulting age is about 3 times as precise as any previous estimate. And it doesn't depend on any kind of fancy modeling. These age constraints are because these are directly connected to many multiple rich and uncontroversial eukaryotic fossil records. And so doing this leads us to the hypothesis that Prochlorococcus, its evolution and the establishment of this niche was really dependent on changing planetary systems in the Phanerozoic, in the Paleozoic, and that likely this niche wasn't even filled earlier. And so the presence of this extensive group of microbes certainly was not there before the Cryogenian, and we cannot use their evolution as a narrative of the robustness of these systems. And likely they evolve in a highly contingent way, sensitive to very particular evolutionary or coevolutionary events. And that's where I'll leave it. Thank you.
Questions?
That was a great talk, thank you.
I know it was a throwaway comment that you used, and I'm sorry to pick up on it.
Yep.
Why do you think they went from being benthic to pelagic in the oceans? What made you think that is that part of the niche filling for these organisms? You had a model where you thought they were in the benthic and then they attach the chitin and then become pelagic. So I'm just wondering where you came up with that hypothesis.
So we came up with that hypothesis because genes for binding and degrading chitin are present in marine Synechococcus and some of the more deeply branching groups of Prochlorococcus. And it's a bit of a mystery. How did they end up being hyper-specialized for this extreme nutrient-limited environment in the ocean? There needed to be some process by which that evolved. They just didn't start there. And so, and in the outgroups on the tree, which I didn't show, are like estuarine and freshwater Synechococcus and Cyanobium and all these other groups. And so there must have been a transition to the marine and then another transition to the oceanic at some point.
Yeah, Anthony Brady here. I noticed on your graph of different nodes and their times, one of them, the bottom of the Picocyanobacteria in general, is right on the cryogenian. And there's of course the older ideas you talk about the Prochlorococcus being related to the cryogenian. Does this like just possibly plop a different node in the tree into being like the same kind of a potential role there, or not?
So potentially, but you'll notice the uncertainties get quite large as we go toward the root because that's further away than those active calibrations with the transfers into the more recently evolved groups. And so I think the uncertainties are still too large for those roots to make any kind of causal inferences, but it would be potentially consistent with that interpretation.
That was a great talk, Greg. I had a question. I think it's really elegant how you really are connecting these genes that we have robust fossils for. I think it's really elegant. Do you have a sense of like, could you make like a list, like a compendium, maybe you have of all the genes like this, not just with these fossils, but even with that? So yeah, like curious about like, you know, is there like a database that could be like, okay, like even if it's very late, like plants or something else that we could maybe infer from, that you could then start building as an idea. I'm not.
Oh no, you're spot on. These 3 gene transfers had already been described in literature, just in surveys of Prochlorococcus, like, oh look, it transferred a gene. So we just found those surreptitiously. But Maya here was just awarded a GRFP, and part of that work is going to be to do exactly what you're describing. So we're developing a computational pipeline to come up with a catalog of these events.
This would be a great resource.
Mm-hmm. That's what we think.
Awesome.
All right, if you have any more questions for Greg, you can ask him at the bar. But let's give Greg and all our speakers one more round of applause.
Thanks everybody. We're having a poster session, a reception now. Stay as late as you like. Any notes about tomorrow? So breakfast is at 7:30. Don't forget you have to check out of your rooms for sessions at 9, and the bus will leave at 1:30. Don't forget we'll have the working group before lunch.
Can you remind people to take their t-shirts?
Oh, if you haven't picked up your t-shirt yet, please do. It's on the table.